First, we'll go over an introduction, then the class articles, then our outside article, then we'll talk about what brings all these papers together, and then conclusion.
To begin, we must first explain biofilms. What are they? Well, as shown by the image on the right, biofilms are microbial complexes identifiable by the sticky substance that binds them together. This matrix allows microbes to share molecules secreted by other microbes found in the film. It may seem like a new concept, but some everyday examples of where we would find biofilms are the plaque found on our teeth or slimy substances found in unclean water bottles. However, the biofilms we will explore today are much different than your everyday plaque. Biofilms are much more complex structures that can infect even the deepest alleyways of our organs. They are often associated with chronic diseases, and they pose a threat to human health due to the difficult nature to eradicate them. In this presentation, we will all explain the issues in treatment of biofilms, the nature of these complexes, and what strategies they use to survive, all by utilizing several scientific papers.
Let's talk about paper number 5, Pseudomonas aeruginosa evolutionary adaptation and diversification in cystic fibrosis lungs. So basically, this bacteria is a highly versatile bacteria that can cause a lot of infections. It's also commonly found in cystic fibrosis patients. Cystic fibrosis is a genetically inherited disease characterized by defects in a transport protein resulting in a sticky mucus, most notably in the respiratory tract. Biofilms are basically cells that are sticking together and another surface. These are adherent cells that become embedded with other substances and make it easier for microbial survival. These biofilms basically create a protective barrier for microbes, especially against treatment. Through patient samples, aka sputum, some data was collected, and this paper discusses phenotypic and genomic studies regarding this bacterium. This is an important article because this paper discusses causes and consequences of diversity of Pseudomonas aeruginosa with respect to the underlying evolutionary processes and the clinical implications. This bacteria must overcome a hostile and challenging environment of cystic fibrosis lungs. The selective forces at play here are antibiotics as well as viscous mucus, oxidative, and nitrosative stresses due to host responses. Hence, antibiotic resistance is a very common adaptation of this bacteria. Overproduction of alginate, which is an exopolysaccharide that plays an important role in development and maintenance of the biofilm matrix, can provide protection to this bacterium. The diversity of CF lungs is the result of adaptative, spatially heterogeneous selection, and non-adaptative, which is genetic drift due to spatial structure processes.
The second paper that we chose was paper 17, Cooperation and Conflict in Biofilms. It's thought that biofilms have high levels of cooperation because biofilms' extracellular polymeric substances are shared between different cells. And it's a shared resource that is produced by one cell, but others can use the resource as well. This article explains and tests if biofilms have high levels of cooperation by using an elaborate individual-based simulation of a biofilm. And this is to look at and find the outcome of evolutionary competitions between strains that have different levels of polymeric production. The model uses a biochemical description of the carbon fluxes for growth and polymer production. And by using these descriptions, it calculates a diffusion reaction effects and the solute gradients that are produced in the biofilms. And this is a new method of modeling, and it's different because it changes how biofilms are expected to respond to changing social conditions. If there are multiple strains in a biofilm, it should promote polymer secretion instead of inhibiting it. So, cooperative and altruistic adaptations show a real problem for evolutionary biology because it's thought that selection for selfishness and cheating should frequently reduce group behaviors. And because the production of EPS in biofilms can be shared by other cells, it would seem to be an exploitable resource. And this is why biofilms are an example of the theory in this paper.
For the outside article, we chose a paper on the survival strategies of infectious biofilms. Its main focus is how biofilm matrix and physiology of biofilm embedded cells influences tolerance. Biofilms are generally difficult to detect and are tolerant to host defenses and antibiotic treatment. They also make the spread of antibiotic resistance easier by promoting horizontal gene transfer. The variety of phenotypes within an individual biofilm is caused by complex 3D structures that consist of many microenvironments differing in osmolarity, nutrients, and cell density. The findings of an experiment performed on Platonic cultures showed that antibiotic tolerance is mediated by starvation, but that high cell density is important for the accumulation of metabolic waste products. The results showed that in the stationary phase, Platonic cells rapidly regain antibiotic susceptibility after diluting the medium. This is because the medium provided nutrients but also protected cell-to-cell signaling. Due to the growth of biofilms, bacterial infections caused by rapid proliferation of Platonic cells have been replaced by chronic infections, hence why a biofilm in growth mode is a risk factor in the spread of antibiotic resistance. Although there is no treatment to eradicate biofilms, the researchers have suggested that using antibiotics beyond their minimum inhibitory concentration and a combination of treatments are the most likely solutions to slowing down biofilm growth. Each article explained how viruses can survive and spread with the aid of a biofilm. Article 5 explained how biofilms help create and maintain the perfect environment in the lungs for CF bacteria. This then results in a very hard-to-treat disease due to the variety of genotypes created. Article 17 explains how multiple strains within a biofilm help promote polymer secretion instead of inhibition, which helps increase the chances of survival for the cells. The outside article describes how the association between pneumonia and the Th2 type immune response helps encourage immunity against extracellular organisms and promotes antibody production.
Let's go over the connection more specifically. Biofilms are of major clinical importance. Almost 60% of current infections treated are considered to have biofilms. In paper 5, we talked about cystic fibrosis lungs and the difficulty treating this disease because of the diversity of bacteria in these lungs due to biofilm development.
Paper 17 discusses the evolutionary conflicts that are predicted over contribution to shared group resources.
Another major point is tolerance to antimicrobial treatment and host defenses. In paper 5, we discussed evolutionary adaptations of Pseudomonas aeruginosa, which includes biofilm development that leads to antibiotic resistance.
Paper 17 discusses how extracellular polymers may provide benefits and antimicrobial resistance.
Finally, survival mechanisms of biofilms, which is biofilm penetration, is slowed down by alginate production by the bacteria Pseudomonas aeruginosa. In paper 5, we talked about production of alginate by this bacteria as an evolutionary adaptation.
Paper 17 discusses how cooperative and altruistic adaptations provide a real problem for evolutionary biology.
After hearing about all the information, we hope you're all now aware of how serious biofilms are against human health. More than half of microbial and chronic infections have the presence of biofilms, and it's getting harder to treat them due to their strong resistances. The reason we chose these 3 articles is because we wanted to discuss the major concern that biofilms pose in many infections. As antimicrobial treatments develop and become more widespread, the microbial world is also adapting to it. Biofilms are making it much more difficult to treat specific diseases, and we use cystic fibrosis and the bacteria Pseudomonas aeruginosa as one of our main connections. Other than just adapting against antimicrobial treatment, the paper showed that biofilms are difficult to remove due to their cooperative nature to survive even in the most stressful host environments. They even end up using host immune mechanisms to survive. Not every host is the same though, so adaptation is very diverse. Biofilms are responding to their different environments, and treatment is becoming much harder. Looking forward, it would be very important to further research and understanding of biofilms. Along with developing treatments, the prevention of biofilms should also be experimented as well. Thank you for listening. Here are our references.